Hi, I'm David Walsh from the laboratory of Stephen Hallam in the Department of Microbiology and Immunology at the University of British Columbia. Today we will show you a procedure for filtering small volumes of seawater for microbial biomass. We use this procedure in our laboratory to study microbial diversity in the marine environment. So let's get started.
Before filtering, first wipe away any condensation from the full bottles and weigh them. Place an autoclave bin on one side of the pump and the waste flask in an autoclave bin on the other. Then place a full sample bottle in the autoclave bin. However, do not remove the cap of the bottle yet. Pass the tubing through the peristaltic pump so that the lower end of the tubing is in the beaker. Using tweezers, remove the cotton from a sterile 25 milliliter pipette and attach the pipette to the other end of the tubing. To begin filtering seawater, open the bottle and insert the pipette. Turn on the pump and set the flow speed to 1. Watch to ensure that water is being pumped in the proper direction. Allow about 100 milliliters of water to pass through the entire tubing. Turn off the pump and attach a Sterivex filter to the male lure fitting at the free end of the tubing. Insert filter into waste flask. Turn on the pump and set the flow speed to 1. Continue filtering.